Hello, let's talk photosynthesis. So the diagrams you're going to see here in this lecture are not going to start out looking a lot like photosynthesis, but we're really trying to get at the question of what is going on, what is driving this process. And it's kind of neat, and it involves a bit of physics. So let's talk photosynthesis as we are going to learn it in this class. All right, so to start off, we've got to cover the very basic physics, which is that, as you know, energy can neither be created nor destroyed, it can only change forms, right? So, what does that mean when light hits something? The light actually hits something, is absorbed, and that actual light molecule, if you like, which is called a photon, ceases to exist in our universe. But it carried a lot of energy inside of it. And so where does the energy go? Well, it can be radiated out in heat or in various other ways. And that's the idea here. Plants aren't just absorbing photons to drive heat, although I've seen a good physicist argue that that's exactly one way that you can think of it. The way we're going to understand it here is we're not going to get all the way into the quantum physics, but we're going to get a little bit in. So this is like a taste of biophysics. What I'm trying to do with each of these lectures is to try and bring you a more sort of fully fledged version of biology, like you would learn it with all the bells and whistles on in one of the areas of biology. And this will help you understand which of those might appeal to you down the road. Okay, so step one, you get a photon that comes in all the way from the sun. Yeah. I guess it could come in from a flashlight. It doesn't really matter. The plant doesn't care. Different colors of light. So each photon is going to have a particular wavelength. Each wavelength is going to correspond to what we would see as a color of light, or there are a lot of wavelengths that are too small or too big for us to actually see them with our eyes. And we call these ultraviolet and infrared radiation, respectively. So what happens in this process is that a photon comes in, and the photon is literally absorbed by some molecules in the plant leaf. And so these molecules are going to take the energy from the photon and they're going to use it. And where this energy goes is it's going to go into one of these photopigments or photosynthesis pigments, a molecule like chlorophyll. And these molecules are set up in such a way that there will be an atom in the center. I think it's a magnesium for chlorophyll type A. And this is kind of the target. And so this is the atom where we are going to direct all the energy from all the photons. The energy comes in, and the first thing that happens is that it's going to take one of the electrons in the outermost shell of that magnesium atom, and it's going to boost the energy level up. So there are a lot of possible, there's, an infinite amount of possible wavelengths of light, I think. And so they would each carry slightly different amounts of energy. But that electron only has so many places that are going to be stable, so many energy levels where it can rest for just a moment and be stable. If you want another explanation of this, the first video that's linked below on Blackboard is going through just the physics on this. Then an electron can get excited and its energy level goes up, and then it's going to fall down to whatever the nearest stable point is. And so the energy level falls down, but of course energy can't be created or destroyed, it can only change forms. And so that energy has to be got rid of in some way. And the way that that happens is we're going to lose it in the form of what we call vibrational energy. So let's see, that we are now on step 3. So step 1 was absorbing the photon. Step 2 was we've absorbed a little bit more energy. It got us past a stable spot, and so we have to lose some energy. And then step 3 is we're losing this energy. Some of this is going to go to what we would call heat, which is a little more complex in terms of the physics behind the thing, but that's the idea. And some of it is going to be lost through vibrations, literally sort of stretching and pulling on atomic bonds. And so we'll lose certain amounts of energy here, and then we are at the stable spot. So think of this thing where I'm drawing the, going across the screen right there. That's our stable position. Now, from there, the electron can remain in this excited but stable position for a little while, but then eventually, and it may be almost immediately, it may take like a minute, it's going to fall back down, it's going to lose its energy, and so it's going to drop back down to the bottom state, which is called the ground state. That's the most basic state where it does not have any extra energy inside of it. But when it falls down to the ground state, it now has to release all of this energy from the top of the this little bar right here. That's our upper energy. That's our energy level where we were stable, down to here. That's our energy level that we've just dropped down to. And physicists can calculate the exact size of these energy levels, but we're just dealing with concepts here. And I only want you to get the concepts. I think that's the important thing for right now. There are more levels, far many, more levels beyond what we're going to do here. But we don't need to do those in Bio 1, I don't think. Okay, so we're going to drop down. We're going to lose exactly this much energy. And now there are two ways that energy can go out into the world. One is that the energy will be converted into a photon. So that molecule absorbed a photon and took all the energy, and the photon disappears itself. It just becomes an energy level in an electron. And then to get rid of that, we basically reverse the process. The electron goes back down to its original unexcited state, and it produces a photon that then shoots off at light speed. So that is one way that we can do this. But a plant doesn't want to do it this way, because if a plant just absorbs a photon and then gives off a photon minus a little bit of heat energy, then the plant hasn't been able to do any work. It has just, it's acted like a not very efficient mirror, basically. So the plant wants to be able to capture this energy and do something with it. That's the idea behind photosynthesis. And so the plant is going to go a different route. And the different route is called resonance transfer. Resonance transfer happens when we can pass an excitation state directly from one molecule to another without any photon being made. The excitement is just passed from one molecule to another. So sometimes you'll hear this described as an exciton, E-X-C-I-T-O-N, transfer. Well, that didn't do very well with the subtitles, but exciton. But we'll just call it resonance transfer. I think that's easier. We don't need to add an extra word to it, but if you see that extra word, that's what it means. It just is another way of saying the same thing that we're talking about here. All right, so let's look at that. Resonance transfer. So resonance transfer means you take one molecule, you put in everywhere that says it looks like HV. What we're talking about there is a photon of light. That's they're Greek letters, and they're used by physicists to reference a photon. So the photon comes in, and it's going to excite this molecule. It's going to excite one of the electrons, which is going to go up to the excited state, and it'll lose a little bit of energy if it needs to. And then the excited state of this electron is going to drop down to original. At the same time, it's going to be transferred to the next atom along. So this usually works best when the atoms are all bonded together in a chain. If they're all linked together by covalent bonds, then you can transfer by resonance energy transfer very quickly. It can also work in between molecules as long as the ends of the molecules are lined up just perfectly. And so in order to make a machine that could harvest as much light as possible, the plant is basically trying to do what a satellite dish would do. You know, those great big bowls that have a little thing in the center? The purpose of the bowl is to catch radiation from all different directions and funnel it into one central point. And that is exactly what the plant is trying to do. It's trying to catch radiation from the sun, being light, essentially, over the entire surface of the leaf, and then funnel that energy into a thing called photosystem 2. So we're going to put our energy into photosystem 2 first, and then we're going to put it into photosystem 1. This could happen in either order and does in nature, but conceptually it makes the most sense to talk about photosystem 2 first, because the leftover products of photosystem 2 are what helps to power photosystem 1. So the two are linked up in this way. So let's talk photosystem 2 first. I think that's the thing to do. And so photosystem 2 is a big collector dish, essentially, for collecting photon energy and directing it toward the very center, a thing called the reaction center. And in that reaction center, there are two chlorophyll, or rather, two pairs of chlorophyll molecules. And each of these pairs of chlorophyll molecules in photosystem 2 is called P680. P680. Yeah, just once though. Okay, how does this happen? That would be a good question. And these are the questions that, drive scientific thought. All right, what you've got here are a variety of molecules which are called photosynthetic pigments. These are all molecules that are able to absorb light, absorb photons, and transfer that photon energy in the form of resonance transfer from one end to the other. And then if there's more than one molecule hooked up next to each other, then that energy can then jump to the next molecule and keep going. So you've got some of these things that you find around the outside of the photosystem, keratines, like an alpha carotene, cryptoxanthin, fucoxanthin. Yeah, and these are going to be pigments that I think absorb in the yellow for them, or they're going to look yellow anyway. They must absorb something else if they're reflecting yellow. And then over here we've got our actual chlorophylls. And chlorophyll A and B are very similar, but they have a little tiny group up here that's different. Other than that, they are essentially the same. And you can see in the center of chlorophyll A, we've got this Mg, which stands for magnesium. That is the ultimate target of all of this resonance transfer. We are going to try to pile photon after photon's worth of energy right onto that magnesium in the center until we get an electron so excited that it basically leaves the molecule. We are going to actually have an electron that's excited beyond the maximum amount it can be excited, and then it just leaves. It is free to go where it will. And that is how photosynthesis is going to start. But we're going to show that in pictures. I want to show you all this a couple of different ways. Each looking at different parts of the process. Okay, so here is photosystem 2. So you'll your eye will probably be drawn to the little cylinders. Each of those is an alpha helix. And you can see down at the bottom, we've got a little region here that's a big long beta sheet. You see the little arrows? So there's our beta, here's our alpha structures up here, like all this red stuff. And yeah, you can see it's mostly alpha helix on the top, and there's a little bit of beta sheet on the bottom. Great. Now I want you to look in between those cylinders. And there you will find the photosynthetic pigments. So you'll have your chlorophylls, you'll have all your xanthophylls, you'll have your keratines. All these sorts of things are going to be lined up. And they're going to be lined up in such a way that excitement, energy, resonance transfer can happen from one molecule down to the end, then jump to another molecule down to the end, jump to another molecule, and this one goes behind this yellow helix, then jump to this molecule, which is then going to go behind this yellow helix, and jump to that molecule, which will then jump down here, which will then jump to this molecule, and then it's in the center. Then it has reached the center of photosystem 2. So it's just like a wire in electronics. These are molecules that are going to act as molecular wires. Only they're not going to transmit electricity by itself. They're going to transmit energy in the form of excited electrons, and they're going to transmit it in one direction, which is right into the center of the reaction center, into that chlorophyll molecule, and specifically into the magnesium in the center of the chlorophyll molecule. All right, so now that we have transferred this energy in by resonance transfer, we are going to do some work with it, do a little bit of work with it. So, first thing, we've got those two chlorophyll molecules at the center of photosystem 2. And there's actually two pairs of these. Each pair is called P680, which is pigment 680, which absorbs best at the red color, which would be 680 nanometers. That's why we call it that. So this P680 is going to be the driver. This is the thing that is going to lose an electron. And this is the thing where all the energy is going to be focused on P680. So P680 can either directly absorb a photon at around 680 nanometers, in which case, great, it's got enough energy and it can go and start the electron transfer chain. Or it can have energy fed in via all those little, the rest of the satellite dish, basically. So if the light lands anywhere on the surface of this leaf and it's hooked up to the reaction center, the light energy will travel in toward the reaction center. That's the idea. Now, the P680 is going to get excited, and one electron in there is going to get so excited, in fact, that it is going to leave the P680 molecule, and it's going to go to a molecule called pheophytin. So where P680 is right here, that's step one. And then we're going to transfer a literal electron, a very high energy electron, to pheophytin. And it's spelled out right down here, pheophytin. I think like fee, fi, fo, fum, like a big giant, you know. And then it's going to be transferred to a bunch of different other steps. And each time it's going to go down in energy a little bit. We're not going to focus on the steps. We're going to focus on the harvesting and what goes on there, because I think there is enough for you to learn and master there without having to go further into it. But like I said, there are lots of areas around this and in more depth in every part of this that are frankly fascinating and where there's still work to be done in all these areas. Okay, so let's see. We've got rid of an electron. And so P680 was a neutral molecule. It had the same electrons as protons. But when it loses an electron's a little minus charge. So you take away a minus charge, and you're left with a net positive charge. And so that's what happens to P680. It turns from P680 to P680 with a positive charge, P680 plus. And that's only because it's lost an electron. See, you've got the little minus next to the electron, so that shows you if you added that minus 1 on the electron to the plus 1 on the P680 plus, you would get back to a neutral state, regular P680. But that electron has too much energy, and so it had to jump off of P680, and luckily pheophytin was just waiting there to catch that electron. It is positioned and held into the perfect position so it can always be there to catch the electron from P680. Okay, now P680 has been turned into P680 plus, and we have basically broken our photosystem too. Because once it's P680 plus, we can't lose another electron. We're just stuck. And so the photosystem has to do the real magic of this, which is it has to find a way to extract an electron from nature to load it back on to P680 so that P680 will then be recharged and ready to go again. And we can keep going. And so this is the genius, I think, of photosystem 2. It's got this little thing down at the bottom. You can call it an oxygen producing center, or you can call it a manganese cluster. There's a cluster of manganese atoms at the center of the oxygen production center. So I like to call it the manganese cluster just as shorthand for the rest. Manganese atomic sign is Mn. And so here you can see here's 2 Mn's and there's 2 MNNs. So we got manganese clusters. There we go. I think each cluster has 4 manganeses, but I think only 2 of them are reactive, if I'm remembering correctly. And so what the manganese cluster is going to do is it is going to supply electrons by taking them from water. So you remember in our discussion of the protease mechanism, remember about oxygen? Oxygen always wanted more electrons, right? It was always willing to throw away an H plus to, go after a carbon or something. It just wants to bond with something. And you notice that in that reaction, oxygen never really lost an electron. It would be given one of its electrons back when we broke a bond, but oxygen never really gave up the electron to anything else because oxygen doesn't really do that. It's very electronegative. It likes having its electrons. That's just how it is. And so to break up a water molecule, so you can get rid of the one H2 and the other H2, And then if you do this to 2 water molecules at the same time, you're going to have 4 H pluses. So all the H pluses are going to get loose. And you're going to have 2 oxygens, and they're each going to have 2 electrons that they were using in bonds with H2. Now, oxygen doesn't much care what it pairs with. It'll oxidize a lot of stuff. And so if you get 2 oxygen molecules with a full set of electrons next to each other, they will try to combine with each other. They'll basically try to stab each other with those electrons. And they'll do it. They will form a bond. But when they form a bond, they'll use 1 electron from the first oxygen and 1 electron from the second oxygen. And each one of those will now have 1 leftover electron that it doesn't need. Those are the electrons that are going to be transferred into the manganese cluster. And manganese is one of these neat metals that can exist in several different oxidation states. And so it's going to go from an oxidation state of 2 to 3 to 4 in a little cycle here. And during this time, it can actually supply electrons to P680. So P680 accepts the photon energy, loses an electron, becomes P680 plus, and then the manganese cluster down here at the bottom, right there, just above the 3, is going to go through its little steps, and the little steps are right here in number 4, and it's going to physically break down a water molecule and release an oxygen molecule. So this is part of why you have to water your plants. They are going to physically, chemically break apart the water molecules. Plants ruin water, which is kind of a neat thing. But in the process, they're going to produce oxygen. And so we benefit from this as animals that are not capable of making oxygen, but that really do need oxygen. So yeah, that's how it works. That's how basically life is possible on this planet. All right, but of course we've only talked about photosystem 2. So what's the deal with photosystem 1? It's essentially the same deal. So in the center of photosystem 1, we have a thing called P700, which is once again 2 of these chlorophyll molecules. And you have 2 special pairs of these, and those are going to be in the center of photosystem 1. And P700 is also going to get excited, either directly from a photon or by light entering somewhere in the transfer chain, and then resonance transfer of that light energy until it gets to the center, in which case it's going to be poured into an electron. The electron's going to get super excited, and it's going to leave, and it's going to go to another molecule. So we have a different electron transfer chain for photosystem 1, but otherwise the principle is pretty much the same. So I think that's fine. If we're talking something on a test, you can just say it basically works like photosystem 2. Except that it's got P700 at the center, which absorbs best at 700 nanometers. And P700 is going to turn into P700 plus once we've lost the electron, because we're going to lose a negative charge, and so we're going to have a positive charge left over. And then the big difference is in the way that it's going to repair its P700. And so instead of breaking another water molecule and making more oxygen, it's going to take the electron. Remember the electron from P680? That thing's been going downhill. So it's been stepping down through different molecules, been passed around, used up, and each time it goes down a little bit in energy, we take that energy and store it in some other molecule, like an NADP molecule, or we store it in molecules that eventually are going to make us ATP molecules. It's not direct, but eventually we do get there. And so then at the end, you've got this electron, and it's no longer in an excited state. It's actually in a very, a bored state, you could say. And the last step of the electron transfer chain is to dump this molecule, this used up electron, onto P700 plus. Because P700 plus is desperate. It really needs an electron to get back to P700 so it can absorb another photon and keep the plant going, keep this reaction moving. And so that's how it works. The electrons from photosystem 2 are fed into photosystem 1, and then the same process happens in photosystem 1. So yeah, the 2 photosystems work together, just like this. So conceptually, we can think of it as photosystem 2 happening first. And then the leftover electrons from photosystem 2 are going to be fed into photosystem 1. I think that makes the most sense. So photosystem 2 is the one that is going to split water and make actual oxygen and H+. And the H+ is what is going to be used to actually drive a machine called an ATP synthase. That is what is going to make the ATP. So we are going to make ATP as a result of this process, but it's indirect. It's another molecule that's going to do that. And we'll look at that molecule in the metabolism chapter a little bit more. So yeah, P680 to P680 plus. The electron is going to come loose. It's going to go to pheophytin and then to a bunch of other plastocyanins and stuff like that. And it's eventually going to drop down, and when it's in the lowest energy state, it's going to be fed into photosystem 1, which just lost an electron because P700 became P700 plus. And so, boop, we're going to feed that electron back in, and yeah, there you go. It's the circle of life. And we get waste gas from the plant, which is oxygen. So this oxygen is basically ruined water. What's left over from water after plants steal 2 electrons from it. They're basically pinning oxygen down, beating it up, and taking its lunch money. Which is why I think photosynthesis is so cool. Pretty much everywhere else in biology, oxygen gets its way. It bonds to whatever it can bond to, and yeah, the bonds are hard to break unless you can give oxygen something. You gotta like bribe it off. But this is just a pure case where we pin a water molecule down, extract its electrons, and yeah, kick the remnants out in the form of oxygen gas. So we're breathing the waste gas of plants. And yeah, by waste gas, I mean literally waste gas. So yeah, very strange, right? And thankfully, plants are going to use some of our waste gas, which is going to be carbon dioxide. So this is what we do to oxygen. We're going to, how we use oxygen. We're gonna end up dumping electrons on it. But more about that in I think 2 sections. For now, we're just concentrating on photosynthesis and just the light reactions. The dark reactions will be the topic of the next one. So here is an overview. So all right. And we're talking in terms of potential energy. And so these are rough potential energies, but they're not far off. This is actually a pretty good diagram. So you've got your water molecule. And we're going to turn the water molecule into H plus and oxygen gas, O2. And in doing so, we're going to start transferring electrons. We're going to transfer them to the manganese. The manganese is going to transfer them to a tyrosine amino acid. And the tyrosine amino acid, which is also part of that manganese cluster, is then going to transfer it to P680 when it's P680 plus. P680, meanwhile, is getting excited by the light, and so it's got an electron that's going to get super excited, and so P680 star, the star means excited, but in our case, it's actually losing an electron, so it's a positive charge. So I think we can call that P680 plus. I think that works just as well. And then we're going to transfer it down to pheophytin and all these other little steps. And there's even iron involved in the middle of that. And we'll drop it down. Finally, this electron is much lower in energy level. Started out up here, now it's down here. We're going to dump it into photosystem 1, which just absorbed some light and went up to P700 plus, again, excited state. And so to get it back down here, that's where we're dumping the electron. So imagine an arrow where we put an electron into this to get it back down here. But yeah, that's how it works. And so then P700 will have its own electron transfer chain, starting with a chlorophyll A0 to a chlorophyll A1 to a bunch of other little things. And eventually we're going to dump all this into an NADP plus molecule, which will then become NADP. NADP is going to be storing an electron, and the plant is going to use that later. It is storing that electron, and you can see it's stored a fairly high energy electron over here. This is the energy of the electrons that we started with, but since we put in so much energy from the light here to boost it up, and from the light again here to boost it up, now we've got a fairly high energy electron that is stored in the form of NADP. And yeah, that will be used later. That will help to power other processes inside the plant. All right, that's the end of this talk. This has been the physics of photosynthesis, or the biophysics of photosynthesis. We left out the equations, we left out some of the heavy lifting and some of the details, but I think this is a pretty good level of detail because at this level of detail you have just enough information to kind of understand why every part of this system works and what drives every part of it. And yeah, the closer you get to actual atoms in biology, the better chance you have of actually understanding what's going on. That's the big take home that I want to give to you today. So, thanks very much.